Hey guys, in this video we will see clot retraction and fibrinolysis. We know that after injury, the break in vessel wall is plugged by platelets. Simultaneously, fibrin is formed by coagulation which strengthens the plug. During the process, a plasma protein called Plasminogen is also trapped in clot. We will come to it later. Now, the clot formation is followed by clot retraction. In this, the clot shrinks with help of interaction of actin and myosin filaments in platelets. This brings edges of damaged vessels closer, which further helps in reducing blood loss. Also, the clot oozes serum. Serum is basically plasma, but it does not have clotting factors as they are used up in clot formation. So, serum cannot coagulate. Next, damage is gradually repaired and normal parenchyma or scar is formed. So now the clot is no more needed and it should be removed. The process starts with tissue plasminogen activator which is released from endothelium. There is also another type of plasminogen activator called urokinase plasminogen activator or simply urokinase. However, that is synthesized mainly during inflammation and is involved in extravascular fibrinolysis. Otherwise, it's the tissue plasminogen activator who is doing the job most of the time. In clot, it binds with fibrin and then activates plasminogen into plasmin. Plasmin cuts down fibrin strands. The breakdown products are called fibrin degradation products. Now without fibrin meshwork, the clot gradually dissolves. The fragments of clot in blood are cleared by phagocytes. And finally we have everything back to normal. Wait, we still have many points to see in this video. Apart from dissolution of clot, plasmin also prevents clot formation in healthy vessels as it degrades fibrin that is inappropriately synthesized in circulation. Next, it is a rather non-specific enzyme and also breaks down some clotting factors like fibrinogen, prothrombin, factor 12, 5 and 8. So if it becomes excessively active, level of these factors might drop, which would hamper normal coagulation. So it is important to put a limit to its activity at healthy sites and mainly confine it to a clot. Let's see how this is done. We know that plasmin initially exists as inactive precursor from Plasminogen. Its activation is dependent on tissue plasminogen activator. However, healthy endothelial cells release Plasminogen activator inhibitor 1. As you might have already guessed from its name, it inhibits Plasminogen activator. So there is minimal activation of plasminogen under normal condition. We also have plasminogen activator inhibitor 2 for this job. However, that is synthesized from placenta and becomes important only during pregnancy. Now, if excessive plasmin still gets activated, alpha-2 antiplasmin takes care of it. It is a protease inhibitor released from liver, kidney and some other tissues. It inhibits plasmin. Thus, plasmin activity is kept under control by plasminogen activator inhibitor and alpha-2 antiplasmin at healthy vessels. However, the story is different where clot is present. Here, tissue plasminogen activator binds with fibrin which potentiates its catalytic activity. So, in presence of fibrin, tissue plasminogen activator can easily activate plasmin. Also, the plasmin thus formed is safe from inhibitory effect of antiplasmin. That is because binding site for fibrin and antiplasmin is same. So when plasmin is doing its job of breaking fibrin strands, antiplasmin cannot inhibit it. Once the fibrin mesh is broken and clot is dissolved, antiplasmin inhibits plasmin and terminates its action. In short, very presence of fibrin increases activity of plasmin, and in absence of fibrin, plasmin activity is minimal. So that was all about fibrinolysis. Now summary. Clot retraction is carried out by interaction of actin and myosin filaments in platelets. It brings damaged edges of vessel together and contribute in hemostasis. During retraction, the clot oozes serum which is devoid of clotting factors as they are used up during clot formation. The damage is eventually repaired and clot is no more needed. So the process of fibrinolysis starts with plasminogen activator released from endothelium. There are two types of plasminogen activators. Tissue plasminogen activator which predominates under most conditions and urokinase plasminogen activator which is released in inflammation and carries out extravascular fibrinolysis. Plasminogen activators convert inactive plasminogen into active plasmin. Plasmin breaks fibrin into fibrin degradation products, leading to clot dissolution. It also degrades fibrin generated inappropriately at healthy sites and thereby prevent unnecessary clot formation. It's a non-specific enzyme and can also degrade fibrinogen, prothrombin, factor 5, 8 and 12. Its excessive activity can deplete these factors and cause hypocoagulability. Now, regulation of fibrinolysis. In healthy vessels, we have plasminogen activator inhibitors. There are two types of it. PAI1 released from endothelium predominates under most conditions. PAI2 is released from placenta and becomes important only during pregnancy. They both inhibit plasminogen activator, so plasmin is not generated at healthy sites. And still, if it's activated, alpha-2 antiplasmin, which is released from liver and kidney, will inhibit excess plasmin. On the other hand, in clot, fibrin potentiate tissue plasminogen activator, so plasmin is generated. Also, fibrin occupy active site of plasmin, so alpha-2 antiplasmin cannot inhibit plasmin. In short, the presence of fibrin increases plasmin activity. That's finally it for this video. Check out more videos at our channel for quick and easy and yet detailed understanding of medical concepts. And don't forget to subscribe because lots more to come. Thank you for watching. See you in next video.